All right, so I think time is there. So let me just kickstart the distinguished Nano ISMB public lecture series. Thank you everybody for coming. My name is Arnold Zhu. I'm from University of Sydney School of Biomedical Engineering. I'm starting senior lectures in the university. And it gives me a great pleasure to be here to introduce the invited distinguished guest from International Mechanobiology Society to talk about the public lectures. So at the beginning of this series, I would like to thank Nano Institute, Nano Initiatives, and also we have the Professor Ben Eggleton to make the open remarks and to demonstrate the support and endorsement to support the mechanobiology research in Australia. So the first talk, I will invite Professor Tang from Department of Physiology at Cheng Kung University in Taiwan to make a short updates about the next ISMB conference. Up to you, Professor Tang.
Thank you very much. So last night, Boris asked me, so what are you going to offer next time? Because we are viewing the eclipse. So this is the answer. So why Taiwan? Yeah, I'm going to show you a few slides very quick. Can I move this down? I have 10 reasons, okay? We just quickly go over. Expensive growth plans, colorful festival, indigenous culture, surrounded by breathtaking nature, the accessibility, and the hiking paradise. So a lot of people like to hike, and the mountain, the ocean in between. You have wonderful place to go, tantalizing test baths. This is, many people know, this is Taiwanese food. Metropolises and the favorite policy. The most important, okay, Taiwanese people are very, very friendly. Okay, and final reason is we are going to celebrate 400 years history of Taiwan in 2024. Thank you very much. So, how is that, Boris? Good? Thank you. Thank you very much. Yeah. And we welcome all of you to come to Taiwan. Tainan, actually. I'm in National Cheng Kung University. Tainan is the ancient capital of Taiwan. Thank you very much.
Thanks, Professor Tang, and also very exciting because after the COVID pandemic, I haven't been using my passport for 3 years, so definitely a great stimulations for us to move on to the next one. So I'm going to my great pleasure and honor to introduce the first speakers, the plenary speaker, Professor Martin Chalfie from Columbia Universities, and he very brief because today we have lots of students and also from general publics attending our public lecture. Very brief. Professor Martin Chalfie shared the 2008 Nobel Prize in Chemistry and for his introduction of green fluorescent protein as the biological marker. But today, his talk title is The Continuing Need for Useless Information. So I don't know, this is how useless it is, but but I certainly can see, we're in the world that with lots of informations. And people talk about their news are fake. There's truth. We don't know yet. But I think I would like to hear Professor Martin Chalfie to talk about this, and it's very important information and how we're gonna apply that to science and also the future of the world. Thank you.
So thank you, Arnold. So what this talk really is going to be about is about doing science very generally. And the title, which does sound a little strange, I admit, is actually stolen from a 1935 article by this man, Abraham Flexner, who wrote an article in Harper's Magazine, The Usefulness of Useless Knowledge. And as you can see on the slide, he says, from a practical point of view, intellectual life is on the surface a useless form of activity. But it is the source from which undreamed of utility is derived. And he also adds, he says, I sometimes wonder whether our conception of what is useful may not have become too narrow to be adequate to the roaming and capricious possibilities of the human spirit. So really what I'm talking about is the importance of basic fundamental research, and not, which has to go hand in hand with any applications. But it's really going to be about this basic research. And in this article, he talks about a conversation he has with this man, George Eastman. Now, George Eastman was the person that started the Kodak Corporation, Eastman Kodak Corporation, film, cameras, and all sorts of things like this. An incredible industrialist. And Flexner relates a conversation he has with him in which he says to George Eastman, George, tell me who you think is the most important scientist, probably 1938, 1937. And Eastman says, that's the easiest question in the world to answer. The person that's the most important scientist is Marconi, because Marconi developed wireless and the radio. And Flexner says in his article that he basically tells Eastman that he's completely wrong, and he says, look, Marconi was inevitable. If it wasn't Marconi, it would have been somebody else, because the real heroes of wireless and radio are the people that came up with an understanding of electromagnetic waves, which are Clerk Maxwell and Hertz. To give you a few more examples along this line, I'm going to show you an infographic that you will not be able to read. That's the infographic, but the citation is there in case you want to actually look at this. It comes from the Quartzoff company. And what they did in 2014 was make this incredible infographic. I'm going to give you a view from the top of it of the iPhone and where all what allowed the iPhone to be made, with its electronics, with its camera, with everything that it did. How could it come about? And if you go from the various items in the picture, internet, LCD computing, battery, and all of that, and you go down a little bit, this is all the science. This is just one part of the science that had to be done in order for that to be occur. And you keep going down and down and down and down, and when you finally get to the bottom, it's discovery of zero, and the first lens, and all the things that we've built on over the centuries, millennia, to finally come up with this final bit of technology. And this is not a new idea, not even for Flexner, because I found a quote from a letter that Mark Twain wrote to Helen Keller, where he says, it takes 1,000 men to invent a telegraph or a steam engine or a phonograph or a photograph or a telephone or any other important thing, and the last man gets all the credit. And we forget the others. It's all that useless information that this was built on. And to bring it a little more closer to home, even now, there was this wonderful quote from an article that was written by Anthony Fauci, in which he said, what is not fully appreciated is the starting point for the timeline of the SARS-CoV-2 vaccines was not the 10th of January, 2020, when the Chinese published the genetic sequence of the virus. Rather, it began decades earlier out of the spotlight. So all that useless information is being used for something else. So how do we get that useless information? I'm going to come up with a couple of ideas, but one is to make discoveries, because we really don't know certainly all that there is to know. So how can we make discoveries? And one way is hinted at by a wonderful quote from the physicist Enrico Fermi, who said, if the result confirms the hypothesis, you've made a measurement. But if it doesn't confirm the hypothesis, you've made a discovery. So how do you make those discoveries? Well, I think the one way is you have to do lots of experiments, because you don't know when these discoveries will pop up in what you are doing. And I'd like to give you a couple of examples from the Nobel Prizes of people that have made these unexpected discoveries. Perhaps my favorite is Robert Wilson and Arno Penzias, who worked at Bell Labs. And at Bell Labs, they had inherited a 30-foot microwave telescope. Then they decided they're going to do some work. Actually, Wilson has described that what it really was is he had a failed PhD thesis attempt, and he wanted to revisit that. So the first thing they did was calibrate their machine. They pointed at the Milky Way, fantastic microwave signal, and then they want to zero it, so they point it away from the from the Milky Way into empty space. And it doesn't go to zero. And they try everything they can to get that reading down to zero, and they fail all the way through until they realize they're actually measuring something. They're measuring the background radiation in the cosmos, which was the first indication of the Big Bang. Completely by accident. So they never actually got a chance to do any experiment. This is just calibrating their machine. And then there's this wonderful quote, or a wonderful thing in John O'Keefe's Nobel Prize speech. He got the prize for discovering cells that respond to position, place cells in the brain. And he says, so one day I'm doing the experiment, and the experiment was to stick a microelectrode into the thalamus. And by mistake, I happened to put it in the hypothalamus. And I found this really interesting cell. And that led to his Nobel Prize. And then perhaps my favorite, and the one that is most related to the work that I was involved in, is the work of Osamu Shimomura. Osamu Shimomura was interested in bioluminescence, how different organisms generate light. And so in 1962, he was working on trying to figure out how this jellyfish, Aquaria victoria, generated this beautiful green bioluminescence. And he did what any biochemist would do. He grinds up organisms, he fractionates the thing, and tries to figure out what he's got. And what he's got is nothing. The experiments fail time after time after time because he just cannot reconstitute the molecules needed to generate light. He's failing completely. One night, after another failed attempt, he's hungry, it's already dark outside, he wants to go home and have dinner. He cleans up after himself by taking all of his samples and throwing them in the sink, where there's seawater and some other things probably. He turns off the light, he's just about to leave, he looks back at the sink, and it's glowing. And he says to himself, well, that's a little unusual, wonder what was in the sink. And he thinks about the seawater, and he realizes that in the seawater, there were calcium ions, and he never had ever added calcium ions to his prep. A couple days later, he repeats it. Sure enough, every time he squirts in a solution of calcium, he gets a flash of light. And he uses that to purify the protein. Now, I've heard of people that throw their preps on the floor, lab bench, and they then work beautifully, but it's not really the scientific method. In any case, he used this to prove that the protein, which he named after the jellyfish, called corin, plus calcium, produces light. The only problem was it was the wrong color. It produced blue light, the jellyfish are green, and so he goes, adds calcium to all the other samples, nothing. And then he realizes maybe it's something that converts the blue light into green. So he takes a handheld lamp and he illuminates each one of the samples, and sure enough, another unexpected discovery, he finds there's a protein that will absorb the blue light, give off green. We now call this the green fluorescent protein. He just called it the green protein. He wrote about it in a footnote. As far as I know, it's the only footnote that won its author a Nobel Prize, but so you don't have to write a lot. And with this added protein, GFP, now there's green light. Accidental discoveries. I happened to hear about Osamu Shimomura's work, not in 1962 or 1989. So it just lay fallow for a long time. And I, at the time, still work on a small worm that is transparent. And we wanted to know where genes were activated, which cells turn genes on. And I've just been told there is a protein that I can see it if I shine blue light on it. I'll see where that protein is if it's green. And I just want to give you some examples. So the idea is the protein works, you just shine blue, you get blue, that it's going to be a lantern in the cell. It'll show us where things are. And working with a graduate student, Geoff Skirkin, and collaborating with Doug Prasher, who had isolated the DNA for this, we were able to first show that it worked in bacteria, and then in the little worm that we work on, showing us the particular cells that we want, and it works in lots of other organisms and lots of cells that people can see. But the real advantage of GFP, which I'll tout for a second, is not so much that you can see it, that's fine, but you can see it in living animals. You don't have to fix the animals, they don't have to be some sort of prepare. You can have the animals make it, and you can watch biology happen. And so here's some movies showing development of early fruit fly, or phase-separated particles in the cytoplasm, something people didn't even realize were there, that they could be visualized. And on the right-hand side is actually watching a mouse cell that's infected with HIV, labeled with GFP, put a virus particle into an associated cell. Watching life happen. But there's been lots of other things that are involved here. We can get this going. Here we go. There have been a lot of unusual uses for GFP. Some of my favorites, there's a group in Japan that decided they were going to put GFP and other fluorescent proteins into silk moths to make fluorescent silk, and they did. I'm not so sure about the usefulness of even this, because I think you have to have a UV lamp that you constantly carry with you so people can see how really cool your clothes are. But another thing, somebody made a laser using GFP, a cellular laser. Bob Berlage put GFP into a bacteria under the control of a controlling region that will turn on in the presence of TNT. TNT leaks from landmines, so this was a way of detecting landmines. Several people have worked on this and confirmed it, but it's still not ready to be used because you don't want any false negatives. So people are still working on it, making that. Eduardo Koch, the artist, commissioned a French biotech company to make him a GFP bunny, that Alba, that was the family pet, and that he would bring to his art shows to get people talking about the connection between art and technology. And GFP is also, if you look at the very beginning of a particular Ang Lee movie, It is the reason this guy is green. Okay? So lots of unusual explanations of this. How do we go about making these discoveries? I think part of it is the idea of asking questions. And I would say there's going to be two parts. One is asking questions, the other is questioning our assumptions. So let me go about questions. So I think I want to give you a couple of examples. When I tell you these, I want to warn you, it's going to ruin your life. You're never going to unthink what I'm about to tell you. But I'd say that questions are all around us. So I'm going to ask for a show of hands. I want to know, so the very first research I did gave me my first scientific paper. I worked on a transparent part of the body. If you know what the transparent part of the body is, raise your hand. This is what I usually get. About 5 to 10% of the audience does it. Now I'm going to give you a hint. It's right in front of your eyes. It's your cornea. We have been looking through the cornea our whole lives, but virtually no one I've ever met, other than the people I happen to work with for this initial research, I certainly didn't, have ever asked the question, why is the cornea transparent? And what's the scientific basis of that? A fascinating, very important problem. How do you get transparent tissue and everything? So that's one of them. Now I'm going to tell you a story. When I was a grad student in a physiology department, we used to teach the first-year medical students about physiology. There was always a lecture in October in which the professor would start talking about the different types of skeletal muscle. And he'd start off and he said, look, there's basically two types. One type is fast twitch. Fast twitch responds very quickly, but not sustained. It's just a short time, so it doesn't need its own oxygen supply. You can go into oxygen debt. But then there's the slow twitch muscle, often used for posture, where muscles have to be working continually, and they need an oxygen supply, and that's provided not by hemoglobin, but by myoglobin. And I can tell you that by this time, all of the medical students were asleep and not really excited about this. And then, now I have to explain, in the United States, we have this holiday at the end of November, Thanksgiving. And so this guy would give his whole talk, let everyone get sort of sleepy and out of there, and then say, and this difference between the fast twitch and slow twitch and the presence of myoglobin, which is like hemoglobin red, is the reason we have dark meat and light meat. In your turkey or your chicken, you will never look at turkey or chicken again in the same way, but he did it so that the students would go home, impress their parents, and the parents would say, yep, they're really learning something in medical school. But these questions are really all around us. I won't go into any more because we have a lot of time. But I want to get at this question of challenging our own assumptions. And for this, I'm going to give some examples of something I liked as a kid. I still like them. These are these matchstick puzzles. And if you've ever seen them, they're things like this one. Here's a pattern of matchsticks. Can you move 3 so you make 3 perfect squares? And you ponder this for a while and you move a whole bunch of things and maybe you come up with this answer. And you get the 3 perfect squares out of it. Or another one of this type is move 3 matches and get the fish going in the other direction. And eventually you might move the matchsticks around and now the fish goes in the other direction. But then there is this horrible puzzle that's given to all of us that do this, and that puzzle is, here's 6 matchsticks, make 4 equilateral triangles. And you move them on the surface, and you keep moving on the surface, and you just start swearing at the matchsticks because you can't figure it out. But that's because you've assumed too much. You've specifically been assuming that everything should be in 2 dimensions. The minute you realize it could be in 3 dimensions, you make 3 equilateral triangles with the 6 matchsticks. But there's actually another way. It's not exactly fair because the matchsticks have some depth, but imagine that they're just lines. The other problem is, why are all the triangles why do they all have to be the same? If the triangles don't have to be the same, here's another way of doing it, where you can have this as well. And then there's the really nasty one that goes like this. Make these 3 matches into 6. You can't break them. Just want to say that. Make them into 6. And you suddenly realize, hopefully, why am I thinking about shapes? Could be Roman numerals. And then you change that again to that. All of these things are that we bring assumptions of how we do our work, what we're thinking about, to our work, and they inhibit us. I had a good friend in my department, fantastic scientist, who went on to head the Neuroscience Institute in China, also has a lab in Berkeley, Mu-Ming Pu. He's made a career of this. He has something he calls the retrospective approach. What we did, one day we were all sitting around talking and we said, Mu-Ming, you come up with so many incredibly good ideas. Where do you get your ideas from? And he said, well, it's actually very simple. I read a textbook. Not anything detailed. I go to the textbook. He went to molecular biology of the cell. He says, and I find something interesting there. And then I ask the question, how do they know that? And he usually finds out that it's a really outmoded experiment, very indirect, and that actually no one knows anything about this. So he designs a new experiment, he tests it with his modern methods, and then he writes a Nature, Cell, or Science paper because he's just changed the field because of what he does, and then he goes back to the textbook to do it again. Really an amazing scientist, but always questioning his assumptions. So I think these 2 things, asking questions, questioning your assumptions, are really very important. And as I said at the beginning, it's the useless information that we build on for all of the applications that we really want. But unless we build up that useless information, that basic science, there's really nothing that we can build on it. So I want to end with my favorite quote about basic science. This was said in 1969 by a different Robert Wilson, Robert R. Wilson, who was called to the US Congress for a meeting, and they were debating in 1969, to remind you, that was in the middle of the war in Vietnam, so very concerned about national security and defense. And they were debating, should we pay for this science fair project, this big particle accelerator? And there was John Pastore, the Rhode Island senator, was the head of the committee, and he was trying to feed easy questions to Wilson so everybody would go along with him. And so he said to Wilson, Dr. Wilson, can you please tell the committee how many ways this particle accelerator will help national security? And Wilson said, none, which was not the answer the senator wanted. He asked the question a couple times, and finally, he says, and I think in frustration, he says, okay, look, in what respect will this particle accelerator help the national defense? At which point, Wilson said the following: it has only to do with the respect with which we regard one another, the dignity of men, our love of culture. It has to do with whether we are good painters, good sculptors, great poets. I mean all the things we really venerate in our country and are patriotic about. It has nothing to do directly with defending the country, except to make it worth defending. Thank you very much.
That is a fabulous talk. Thank you so much. First of all, I would like to see if anyone from the audience have some questions. I'll just pass this.
The first part of your talk deals with that famous quote by Isaac Newton, who famously said, if he'd seen further, it's because he'd stood on the shoulders of giants. Was it that you were alluding to in that first part?
No, I was alluding to Abraham Flexner. There is a problem with the Newton quote. There is some debate about that. It's often used as what a wonderful man Isaac Newton was, to not take all the credit and to say, I have worked because of what I have done is because of others. In fact, it was a letter to John Hooke, Robert Hooke, and who he did not like, and who had scoliosis or some other back problem and was sort of bent over, and so was not a giant. And I think that there's some people that feel that what Newton was saying was, I stood on the shoulders of giants, not shrimps like you. So it's a little bit hard to go into that about what it actually is. There is some debate. Did he really mean it? Didn't. Modern day, we always think about it like, oh, what a wonderful thing. He was not always the nicest person in all of this.
Now, my second question actually gets back to Mu-Ming Poo. There's a famous Australian, in fact very famous, Dr. Barry Marshall, who proved that stress does not cause ulcers, and his view was it's called by bacterium. Now, to prove this, he actually took the bacterium himself, got the ulcer, took antibiotics and cured the ulcer, and I believe, like yourself, he also got a Nobel Prize in physiology.
He did share the Nobel Prize for that work, yes. So, absolutely. I'm not advocating that everyone should eat their own experiments, but sometimes it works. Any other questions?
Great talk. Before lunch, I asked you what you made of Donald Ingber's concept of tensegrity, and you said it was leading to something. So it'll meet your test of being useless information unless we know where it's leading. Can I suggest that it explains the architecture of the universe and the architecture of human behavior made up of complementary, opposite, paradoxical characteristics?
I think this is a longer debate than we really have time for. I think that he did direct people towards thinking about the mechanical features of cells, and I think that was important. Whether all of it was right or not, I think there's some debate. But anyway, any other questions?
So Marty, how do you get funding agencies to want to fund the work to generate useless information?
Okay, so this is a fantastic question, and I'm going to explain. So if this takes too long, just kick me out of here. So what usually non-scientists, Rong understands all this, but what non-scientists usually think of when they think about funding is a model that comes from business, which is a contract. I promise to do A, B, C, D, and E, and if you like that, pay me, and I will deliver on A, B, C, D, and E. That, however, is not how, for the most part, we are funded for our research. We are funded by grant proposals, and the grant proposal basically says, in my opinion, right now, when I look at what is the most important series of things to do in this field, they are A, B, C, D, and E, and I'm going to start to work on that. And if you think that's right in the way I'm doing things and what I've done in the past, fund me for that. But then there's the unwritten part of the grant. And that unwritten part says, if someone invents GFP or CRISPR-Cas9 or some other way to do my experiments better or to ask a better question, or if I make that discovery of something I wasn't expecting, I have the support to go after those things. And then when I renew my proposal, you can decide, did I make the right decision? Did I actually make the discovery? So I'm not saying just go off and do whatever you want, but we do make these discoveries, these unexpected findings, all the time in our research. And we have to decide, are we going to go down this path or are we going to go down another path? And so it's, right now in the United States, there's a very nice type of grant from NIH, General Medical Science, which is called a MIRA grant, which is basically a very short proposal that says, what did you do in the last 5 years? And what are you hoping to do in the future? But don't give us the details, give us the general direction, because you've accomplished so many things in the past, we're gonna let you continue. And as long as the next time you apply, you've also made more discoveries, more headway in the thing, we're gonna support you. So they support the people rather than supporting the project that a contract would have. So I hope that answers your question.
I think there's one more question.
Thank you. That was the question I was gonna ask, but I have new questions. I'm still at my early stage of research career and haven't yet made accomplish like you. But what I was thinking is, because you know, it's really important, the basic research, but you will make a lot of, I mean, not you, but people who go into a lot of wrong direction and not generating, you know, findings that was publications, but that all take time. And at the current environment, they kind of give you limited time to do your research work, to generate publications. Do you have any recommendation, like suggestion or advice to, you know, emerging researchers? How do they balance this, or how do they advance their career, in particular in basic research, when, you know, sometimes your hypothesis was wrong and you can't publish?
So there's a couple of things here. Sometimes I've been asked by students, how can the heads of the lab be so happy all the time, but I'm miserable? Because my experiments are working. And I'm going to actually get to the point on this. And that is, I feel that the reason the heads of the lab are happy, but maybe there's some individuals that are not in the lab, is because the heads of the labs have more than one individual that they're talking with, and somebody's work is working. And so that's why you're happy. Now, I'm going to take that and say something you're not going to want me to say, and that is, do two things. Because if one's not working, maybe the other one is. It's when you butt your head against the wall doing one thing that it becomes frustrating. The other thing I want to say is I want to talk, my daughter now is 30, but when she was 12, she and my wife went with me when I was inducted into the National Academy of Sciences. And when that happened, if any of you have trouble sleeping, get a movie of the induction ceremony of the National Academy of Sciences. It is unbelievably dull. You have to realize there's like 70, now 100 people that are being awarded this. And as they're being awarded this, in the audience, there's maybe a few of their family members. They're interested in that person, not in any of the other people, right? So you got to listen to all this other stuff. You go on the stage, you shake hands with the president of the National Academy, you have a picture taken, you walk across the stage and sign a book. But they say one or two sentences. And I figured, oh, my 12-year-old is probably having a fit because she's been made to sit through all of this. So I finally get down to her and I say, sweetie, I'm really glad that you're here, that you and Mom are here to help me celebrate this. But you must be bored out of your mind. And she looked at me, as she often looks at me, that I am the stupidest person on the planet, and says, Dad, don't you understand about these people? So I played along and said, well, what about these people? She had been listening to those sentences. And when she did, she said, Dad, every one of the people on that stage were the first person in the world to see or do what they did. The very first person. Now, to me, that was a profound observation from a 12-year-old, but it also points out why we fail. We are always trying to learn something that no one else has learned yet. And we're not good at that. And so we're going to fail. And so we have to keep trying to do different things to be able to get that to happen. But that's the reward is when we are the first person in the world to see something happen. It's pretty exciting. That's the give and take. But it's inherent, I think, in what we do that experiments will not work every time. And it's something that we have to live with. And if people aren't comfortable with it, probably not the right job for them. Anyway.
Thank you so much.
Thank you.
Just conscious of time, I'm gonna just pass the this discussion panels. Because Rong already asked questions. I will move on to the next speaker, Professor Michael Sheetz. Oh, you might.
Okay. I believe it's on. Yes, thank you. So following after Marty, who used to be a colleague of mine, my approach, I must admit, is different. I would want to pick up on his last point and say that a lot of what I'll be talking about today has to do with serendipity, which is to say that we don't know what's going to happen, and all of a sudden, when we put A and B together, we get an unexpected result. It's indeed those unexpected results that are the basis of what I'll be talking about today. And I'm going to put it into the context of the mechanobiology, because that, in a way, is a sort of underappreciated aspect of biology. The genomics and biochemistry revolution has brought us a lot of tools, but I maintain if you spread out the parts to an automobile up here, you won't know how it works. So it is indeed moving from that parts list to how the organism functions that is a major challenge from here on. And the parts that I'll be talking about today are some unexpected findings that rely upon mechanical perturbations of cells to, one, inhibit tumor cell growth, and two, to cause rejuvenation of senescent and aged animals. So, and as I say, these were unexpected findings. So, by way of background here, what we're suggesting is that this organism, Rajiana from Papua New Guinea, north of here, is built by single cells. And those interactions are genetically programmed such that another bird with essentially the same genetic makeup looks the same. And yet inside that cell, the actions are all driven by diffusion. Even myosin moving on actin relies upon diffusion and ATP biasing that diffusion such that you have movement. And so it is an isothermal machine we're dealing with. And diffusion is by its nature stochastic. And somehow nature has derived a way of taking that thermal energy and converting it reproducibly and deterministically into a way of doing things. So we don't understand that. Further, if you look at the organisms, they are not only able to form the original organism, but they're also able to repair it upon damage. Best example is the salamander. You can cut off a limb and it will regenerate. Humans aren't quite as good. It turns out if you can cut off your end of your finger here, it will grow back if you are under the age of 1. Not recommended, but something which we have a limited amount of regeneration. Okay. So, but the other important aspect of regeneration in terms of cancer is that you require the organism now to activate growth of otherwise differentiated and adult cells. So you have to push them back into the growth cycle. And what that does for you is that opens up a window for continued growth. And so when you go around this cycle here, you have injury, you remove the block to growth, the tissue grows, and you have injury repair, and you're back to where you were. But in some instances, you have continued growth, and that's cancer. And most of them are not a problem. I used to go sit out in the sun, so I have many of these nevi, which are small cancerous growths which don't amount to anything. So, but in certain instances, you add additional features which suppress apoptosis, telomere elongation, etc., and you have tumor growth. And metastasis sends it around, those cells are able to divide, and so forth. So the real question is, what is it that removes this block to growth? And it seems that this is interestingly enough, a deletion of a sensor in those cells. And that sensor is involved in sensing rigidity. So it turns out that many aspects of how cells determine what type of cell to become rely upon the mechanical aspects of the environment. This was highlighted by Dennis Disher, who was part of this conference. And what he found was he took mesenchymal stem cells, bone marrow cells, put them into different environments, and they became different cells, different cell types. On soft, they went down to a brain, on intermediate to muscle, and on rigid to bone. And so that meant that they reacted to and responded to that rigidity. On the other hand, when you look at the early studies of cancer, what they found was that when they took tumor cells out and grew them in the test tube, they would love to grow in a variety of different environments. And in particular, they would grow on soft agar, and the normal cells would not grow at all on that surface. So it's this growth on soft agar, i.e. a non-rigid environment, that distinguished. So the question becomes, how then do the cells measure that environment at a nanometer level, subcellular level? And in the area of physics, there's a lot of interest in the mesoscale, not the atomic scale or the macroscopic scale, but that intermediate scale where you have hundreds to thousands of molecules and you have an intermediate type of behavior. Similarly, in biology, we understand a lot about the structure of proteins at the angstrom level, and we know how cells behave, but we don't understand a lot of the intermediate levels. And here what I'm showing is a cell spreading on the surface, sensing the rigidity of that surface by pinching that surface. You and I can measure rigidity by pinching an object. The way the cell does it is by pulling to a fixed distance, and the force that's generated then is a measure of rigidity. And how do we normally pinch things? It's driven by myosin and a muscle sarcomere. So when we look at this structure, what we see is that when that contraction occurs, here we're using nanometer level pillars of PDMS soft rubber, and they're being pulled together by the cell, hence the arrows moving toward each other. And if you look at that displacement versus time, you see that it is indeed just like a pinch. Pulling to a constant distance, and you measure the force. And the force tells you what the rigidity is. And sure enough, we find myosin in the middle here and actin on the other side. So it's like a muscle sarcomere. So nature has taken what is the basis of muscle and moved it now to another use. Took this tool and put it in another situation. And if you lack that tool, the cell doesn't know anymore whether it's on a soft surface or not, and it becomes like a tumor cell and grows on that soft surface. And so what we found is that there are 8 or more proteins now that individually, when they're depleted, will cause the loss of that property of rigidity sensing. And you find a significant number of these are what are called tumor suppressor proteins, which means that in their presence, they suppress tumors, but tumors grow when they are depleted. So we suggest that, or at least I suggest, that what this means is that they are part of this sensory apparatus that keeps the cells from growing on soft surfaces. And sure enough, this one in particular here, DAP kinase, death-associated protein kinase, is the protein which activates apoptosis of normal cells when they are put on soft surfaces. So what this says is that these cells are in a different state or behavior, tumor cells, that is. And the question is whether or not the properties of those cells are changed in a way that allows us to particularly target them. And what we found quite by happenstance is that if we mechanically distort those tumor cells, they would die, whereas our normal cells would grow better. And a colleague suggested that we could do that distortion using ultrasound, and we tried it, and sure enough, it worked. So here we see the growth of tumor cells in a chick choriontonic membrane, where the tumors grow based upon the hormones and nutrients from the chick before it gets an immune system, and you see nice growth there. But if we treat with the ultrasound, we kill it. So it's saying that mechanical distortion now causes an inhibition of tumor growth. Now, does other types of mechanical distortion also block tumor growth? And this is the model of how it happens. We won't go into the details of that. The important thing here for this audience is that you get the same type of effect as the ultrasound with mechanical distortion from exercise. And that the NCI lists now these 7 cancers which are benefited by, or the patient is benefited by exercise, and 8 more. And I submit to you, it's indeed the mechanical distortion and the mechanical sensitivity of the tumor cells that's important. Okay. And this all started in my lab because we were using distortion to grow cells on soft surfaces, and it turns out we can do that. Okay. And then we tried to see what would happen with the tumor cells, and the postdoc came back and said they die. So that led us then to get a better understanding of how these tumor cells behave. Now, this was all done while I was in Singapore, and we moved to America, and right before COVID-19 started. We've been isolated there, and this is one of our first trips out. It's wonderful to be here. But a postdoc joined the lab when we started there, and he had been working for his thesis back in India on taking mesenchymal stem cells and expanding them. And they would only be expanded through 9 or so passages, grow it up, and then you take EDTA and protease, and then you take the cells up, dilute them, passage, so forth. And so it was limiting. And when he came to the lab, I said, well, why don't you take some of those cells that wouldn't grow, and we'll treat them with ultrasound, just an idea. We didn't know what would happen, and he came back to say, they're growing. So we followed up, and we found that senescence cells were being rejuvenated by the ultrasound. Okay. So I'll digress here to talk about, indeed, what's happening with aging and what is senescence. Because it seems that, and I truly didn't know anything about senescence when I went to Galveston. So it was a totally new area to me. So cellular senescence in this circle of what happens with aging is only one part of it. However, as I look at this issue much further, it seems that what we experience when we age, and I'm including myself in that, is the presence of the senescent cells. And what does that mean? Senescent cells don't grow. They have increased this beta-galactosidase activity as a marker, and they produce and secrete to the environment this senescence-associated secretory product or phenotype, which is SASP. And SASP feeds back on healthy cells to cause them to become senescent. So as we age, then it's a downhill spiral, okay? Because the more senescent cells you have, the more SASP, and the more normal cells are converted into senescent cells. And it doesn't matter whether you have DNA damage or reactive oxygen species or other types of damage, that all leads to cell senescence, okay? So how are we going to get around this? Okay, this is sort of the diagram of what's happening as we age. These purple cells are the senescent cells, and when an organ reaches a tipping point, you have too many of those around, it shuts down. And it's that negative feedback cycle that we all are facing. And what can we do about it? Okay. One approach the biochemists are taking is called xenotherapeutics, where you take a drug that causes those senescent cells to die and you give that to the individual. It seems to work a little bit in mice. And the reason that we think it's just the senescent cells that's important is illustrated by this experiment, okay? Where they've taken in mice now and put in human pre-adipocytes. And they've put in adipocytes that are normal or ones that have been irradiated and are senescent. They wait 2 weeks and they measure physical functions of those mice. And the mice with the senescent cells, they're not muscle cells now, but they're just the presence of the senescent cells in the body, perform less well on the treadmill, on this inverted hanging assay, and on the grip strength. So those interfere with our performance, okay? So if we can treat those with ultrasound and reverse it, maybe we can reverse aging, okay? So here's an experiment where we looked with a variety of agents to produce senescence. The cells after treatment don't grow. Their cell area is huge. They all produce this SASP that negatively feeds back. And now when we treat them, they will be rejuvenated. But in terms of the mechanism of senescence, it seems like the cell is hoarding this material because if we look at the size, their size has increased dramatically. And so now when we go and treat with ultrasound, what we're doing is activating the house cleaning mechanism of the cell, namely autophagy. And now the cells will grow, they decrease in size here, and they no longer produce SASP. So here we see the growth rate is higher than even the control cells before they became senescent. The senescent cells don't grow at all. Okay. So what can we do with that? In a practical way now, we can take mesenchymal stem cells, what this postdoc was working on before, and we can continue to expand them. And so we can use autologous stem cells, expand them enough to cause repair, and they retain their phenotype. So they, after this expansion, they'll still go down and produce adipocytes or produce bone cells if subjected to the right environment. Okay. So that's useful. Okay. What happens in this is that we see with ultrasound a dramatic change in the morphology of the cell. One of the ideas of why this works, or why the cells become senescent, is that their mitochondria fuse together and they have this block to autophagy, which causes a rise in their lysosomes, both of which are reversed by the ultrasound treatment. Okay. So it seems that that block to autophagy is what the ultrasound is doing. It's hitting a complex called mTORC1, which is on the surface of the lysosomes, and the ultrasound is likely distorting the lysosome and thereby producing that inhibition of mTORC1, which leads to autophagy activation. Okay, the question is whether or not this will have any practical benefit at the organism level. So what we've done here is to treat mice. These are mice that are almost 2 years old, the normal lifespan of the mouse. We're treating them with ultrasound and asking the question, will it affect their performance? And we find that with the performance assays, it does work. And that further, these mice were scheduled for the euthanasia because dealing with old mice, the people come around and inspect the cages, and if the mouse is on the edge of death, we're not able to perform experiments on it. These, however, we had special permission now to treat them, and their activity improves, and they're alive today. So it seems that this ultrasound is indeed working, and the question is, why? And I suggested earlier that the senescent cells are the reason for the problems. So are they now reversing the senescence of the cells? And indeed, what we did was to take out the pancreas and the kidneys from these mice and to measure the level of the various senescent markers in them. And sure enough, there's a factor of 3 or 4 decrease in the level of senescence in these cells, in these tissues. In particular, with kidney, we see it going from about 70% senescence to 10 to 20%, likewise with the pancreas. And this can easily explain why we have type 2 diabetes as we age, because the fraction of senescent cells in the pancreas is very significant. So this leads now to the idea that what we might want to do is to ultrasound treat humans and thereby perhaps reverse this. We're in the process of getting IRB approval for this and for treatment of diabetic foot ulcers. But as I say, getting back to Marty's points about discovery, this came out of a serendipitous experiment where the postdoc was bringing in a system from his thesis, and we had something going in the lab. We mixed the two and got an unexpected result. And that's where we feel progress comes, and that's what makes life in this so much fun. And this is the credits that I have for all of this, and I thank you very much.
Thanks.
And by way of disclosure, I am forming a company to try to follow up on this. Okay.
Just really quick, for those, I missed the introduction at the beginning. For those who haven't known Professor Michael Sheetz, he's a professor at the University of Texas Medical Branches in Galveston. According to Mike, there are a lot of beautiful birds there, and that's a very beautiful place. So I'm going to pass the question to Matthew.
Thanks for a great talk. I was interested in lifespan across different animals. There was work earlier this year that showed that the somatic mutation rate is correlated with lifespan. So there seems to be some evolutionary limit on lifespan across different mammals. At the mechanistic level in senescence, do we know much about how that affects in different short-lived and long-lived animals versus us? Which of those mechanisms are ramped up or ramped down?
Well, I mean, the point you raise is that indeed the genome might be the critical aspect of determining lifespan. Okay. It certainly wasn't back in the 1850s, and the big difference between then and now is hygiene. Okay. So I don't know that there's an absolute limit, and I don't know if in this situation the ultrasound reversal will be limited by genetic damage. Okay. So that's where we stand right now. Certainly, DNA damage does lead to cell senescence. Okay. So that would push one down that pathway. But whether the genome is damaged so much that you can't rejuvenate it, we don't know. And the suggestions from what we've seen so far is that the amount of damage to the DNA is not the limitation in this situation. So it's an important point, and I agree with you. We're indeed looking into that now. Okay.
Thank you.
How do cells in a body know the age of the host?
How do the cells in the body know the age of the host?
To create this aging process and disposition to degeneration.
The issue that seems to be, is illustrated from those mouse studies, is the gradual increase in senescence. Okay. It doesn't, no. But the issue is that we experience damage all along, and as we get more and more senescent cells, those positively feed back and create a problem. What counteracts that is exercise. So we can, feel much better, have a better quality of life, and have fewer senescent cells by exercise, distortion of the cells, which is basically what the ultrasound is doing.
How does the clock work? How does a body know its age?
The body is, when you build a machine, it knows its age from its use. And likewise, we are being used and we accumulate damage. And we accumulate oxygen radicals that cause protein aggregation, protein cross-linking in collagen, etc., all of which tends to make the organism less flexible and more prone to senescence. Okay. So those are happening with time.
Yeah.
Yeah.
Thank you for a very nice talk. Actually, my question is, because you mentioned before the different type of cells in different tissue, they have different mechanical property. But in terms of you use ultrasound to kind of change mechanical property, my question is different cell type, do they need different dose to kind of modify some kind of carrier range or mechanical property?
That we understand relatively little about at this point. We do know that if we treat the cells in vitro with the ultrasound, that's different than treating in vivo, and that if we put the cells on soft surfaces, the ultrasound has a different effect or different max ideal power range than if they're on rigid surfaces. So indeed, it's affected by the organization of the cell cytoplasm. And so we just don't, at this point, fully understand. Okay.
The senescence that you alluded to, is that perhaps a metaphor for that children's game where you've got a group of people, say 20 people on one side and one person in the middle, and they have to run from A to B. Now, as they're running from A to B, the person in the middle has to tag somebody. Now, once they've tagged somebody, they join that person in the middle. So eventually, you've only got one person running across, and you've got 21 people tagging them. Is that perhaps a metaphor for senescence? Well, that's my first question.
Yeah.
My second question was Nietzsche. Nietzsche famously said, that which doesn't break me strengthens me. So are you perhaps suggesting that Nietzsche's perception back in the 19th century is a version of the benefits of exercise? And my third observational question is, does ultrasound and exercise have the same benefit by a different medium? And have you considered putting the two together? In other words, exercise plus ultrasound. So that's sort of three easy questions.
Okay. So in the first case, yeah, I mean, you're describing a positive feedback type of mechanism. And indeed, senescence is depending upon your point of view, a positive or negative feedback situation. Okay. What I didn't show was our results that when we ultrasound treat normal cells, they secrete components that activate growth of senescent cells, i.e. the counteracting type of behavior, okay? So that if you exercise and you produce those components, then you will decrease senescence. And many describe exercise as a senolytic, which, increases your behavior, positive behavior, and so forth. Okay. So, and one of the reasons why we focus on the spa as opposed to treating just an organ that may be particularly senescent. Okay. And I've forgotten your last question. What's that?
Nietzsche's famous quote, that which doesn't destroy me strengthens me.
I mean, I prefer the use it or lose it, but the strengthening is indeed, what we're talking about here, I would describe rather as reversing that downhill slide and to bring them back into the normal range. So what we're suggesting is that the ultrasound can improve quality of life. the mice are behaving, like they're having more fun, they're exploring more of their cage, etc. after ultrasound treatment. It's not that they have new capabilities, but they have a quote-unquote better quality of life.
All right, so I would say thanks, Professor Michael Sheetz again for the public lecture talk. Thank you very much. So just conscious of time, so I will move forward to our third speaker and also the last speaker, Professor Rong from Mechanobiology Institute in Singapore University. And then Professor Lee came from the Johns Hopkins Universities, and she served as Director of Center of Dynamics at Johns Hopkins, and now she is the Director for the Mechanobiology Institute at the Singapore in US. So I'll give this to Professor Lee.
Thank you, Li-Ning, for your introduction and kind invitation. So I'm going to start by actually giving a little introduction to Mechanobiology Institute, or MBI, at NUS. So MBI is one of the one of several Singapore's National Research Center of Excellence. It was founded in 2009 with our founding director being no other than Professor Mike Sheetz. I took over about 3.5 years ago, so we're now a research institute focusing on mechanobiology with about 150 people, including faculty, staff, students, and postdocs. And we have a fantastic open lab infrastructure and also 4 fantastic core facilities. So we really enjoy doing science together, pursuing interdisciplinary and highly collaborative work. So if you happen to be in Singapore, welcome to come and visit us. I prepared these slides in case there are people in the audience not familiar with mechanobiology. So what is mechanobiology? As I just mentioned, it's a highly interdisciplinary area of biological research. A broad perspective for mechanobiology is that it focuses on biological systems from a physical and mechanical perspective, rather than from a chemical or genetic perspective. So one of the very important concepts that we all think about is forces. So for example, how are forces produced to drive form and function across skills, like Mike just discussed. And how are forces or mechanical cues actually play regulatory roles in biological processes. So these are some of the fundamental questions that we hope to address. And recently, we have expanded our fundamental research to ask questions related to aging, organismal aging. How do, for example, biomechanical systems age, and how these changes during aging can lead to age-associated diseases. So we just had a discussion about aging and senescence. Here's another way of thinking about aging. So if we think about cells and tissues as these little machines, if you think about the machines or cars running on the street, then aging is really a loss of balance between wear and tear and damage, and the ability to repair and regenerate. So our cells and tissues actually has enormous ability to self-repair and regenerate. However, if that process during the aging process cannot keep up with wear and tear, then the unhealthy states start to emerge. So what's really fascinating to us is that mechanics or forces actually play dual roles in these processes. So you can think mechanics can cause wear and tear or damage. Like, my knee is still having a teared meniscus just from jumping or running around. But at the same time, mechanics also provide the signals and the mechanisms for repair and regeneration. So we want to understand the role of mechanics still in these processes, and hopefully we can help ourselves to retain this balance and to prolong a healthy lifespan. So now I'm going to tell you about And that is loss of proteostasis, which is short for protein homeostasis. And the consequence of that is protein aggregation. So just as a way of kind of review, where's the pointer? There, I guess I'll just use the pointer here. Proteins, as this one, can we see? Oh, it's tiny. Maybe I'll just point here. So proteins are made through translation in the cytoplasm, and after a nascent polypeptide is made, it has to be folded into the native conformation in order to be functional. For example, to act as enzymes. Now, folded proteins can also become unfolded or misfolded, especially under stress, and I'll tell you more about that. And misfolded proteins can either get degraded, or if they accumulate, they can form these aggregates. So you can kind of imagine protein aggregates are these milk curds forming in a spoiled milk. And there, the stress is actually the low pH when the milk is being spoiled. So you can imagine how nasty it could be if your cell is full of these aggregated proteins. And in fact, protein aggregation is a very common hallmark of age-associated diseases. And you're probably more familiar with neurodegenerative diseases such as Alzheimer's, Parkinson's disease, and this motor neuron disease called ALS. And these diseases all have aggregation of different specific proteins. And in addition to neurodegeneration, now there's emerged more and more evidence suggests many other age-associated diseases such as myopathy, heart failure, type 2 diabetes have all to do with protein aggregation. Now, how do we think about protein aggregation? It usually reflects there's a disruption of proteostasis, and there could be many underlying causes. So proteotoxic stress is a direct cause of protein aggregation. Can be a direct cause of protein aggregation. Protein toxic stress referring to the kind of stress that interferes with protein folding. It could also due to a loss of balance between protein production and protein turnover. And finally, a lot of other kinds of stress not directly associated with protein folding, such as genotoxic stress or metabolic stress, have also been implicated in protein aggregation for reasons we don't quite understand yet. So to study it, so now because protein aggregation has been implicated in so many different types of cells and tissues, there are lots of places one can study protein aggregation. But in our work, we actually started with a very simple system, or a simple kind of cell. It's actually a unicellular organism called the budding yeast, or Baker's yeast. Its official name is Saccharomyces cerevisiae. It's the same kind of yeast that people use to make wine, to make beer. I'm very proud of our little organism because in the past 15 or so years, there has been 4 Nobel Prizes given to scientists working with yeast to make fundamental discoveries in processes from cell cycle regulation, telomeres to autophagy that Mike just mentioned, and vesicular transport. So it's a really powerful system for biologists to discover the most general principles of relevance to eukaryotic cells. So the reason that we were using yeast cells to study aging and protein aggregation is that yeast actually makes aging really simple. So here's a mother cell, here's a yeast cell. The way yeast cells give birth to another cell is to generate a bud, and then segregate the genome, duplicated genome into this bud, and then this bud comes off, so that's a new cell. And then this mother cell can bud again and generate another daughter cell, and so on and so forth. But this mother cell cannot go on forever. It actually undergoes replicative aging. So after about 20 to 30 times of giving birth to a bud, this mother starts to get old, they start to get wrinkly, and start to divide slowly, and then undergo senescence and death. But what's really fascinating is that even for an old mother cell, when she gives birth to a bud, this bud is always a newborn that has this whole full reproductive potential. Okay, so what this means is that at each time, each cell division, each time right before cell division, what makes a cell old and what makes a cell young have already been segregated to the two sides of the different sides of this cleavage plane. So this offers a really nice system for us to try to understand why, what are the determinants of cellular aging? What are being segregated? How are they segregated? And finally, we can even use this system to ask, why do cells really need to age? Why can't they just be immortal? I'm not going to have an answer for that last question, but I'm going to tell you about some of the things we've learned about aging determinants and their segregation. And this determinants that we're going to talk about today, and there are other things that are also important, but I'm going to focus on protein aggregates, because protein aggregates actually also naturally accumulate in the aging mother cell. But what's really fascinating is that when this aging mother cell, after accumulating these aggregates, when she gives birth to a bud, she really keeps all that bad stuff to herself. So that allows the bud to be born with a clean slate. So how does that happen? So the work started with, in my lab, along this line of work started with a graduate student, Chen Kai-zhou. And Kai was just playing with protein aggregates. He was inducing them with heat, with different kind of stress, oxidative stress. He was inhibiting proteasomes. He made an unexpected observation that every time he induced protein aggregates, here actually these aggregates are visualized with a protein that binds to the surface of these aggregates and also have a tag that is green fluorescent protein that Marty just talked about. So you can actually see they're enormously useful that help us visualizing structures and protein localization in live cells. So these green dots here, foci here are aggregates, and the red is mitochondria. And just as a way of reminder, so mitochondria are cells' powerhouse. It's where cells use oxidative phosphorylation to generate ATP. Another thing I just want to point out is that the textbook version of mitochondria are always these little peanut-shaped beady stuff. But actually in the cell, mitochondria are often in these nice, beautiful long tubes. And mitochondria can also undergo very dynamic fusion and fission. So you can also see some parts of mitochondria are these little structures, others are these big long tubules. So, and then Kai made a movie asking what happens during cell division. So what you're gonna see here's the mother cell, has aggregates in green and mitochondria. And here's where a bud is gonna come out. And the mother needs also to send a bunch of mitochondria to the bud because the bud needs mitochondria also in order to live. And so you can see that you won't see the bud, but you're going to see this mitochondria going into the bud. So the bud will inherit its own stash of mitochondria, but aggregates don't follow. So the aggregates got captured by the mother mitochondria and prevented from going into the bud. And in fact, we now have a hypothesis that we think the mitochondria that go into the bud are not only damage-free, they're also more or less the newly synthesized mitochondria, whereas the mother cell keeps the damage associated with the old mitochondria. Now, but the mother cell does not just passively keep the aggregate on her mitochondria. Actually, the cell can actively dissolve these aggregates. So for example, here's a movie of a bunch of aggregates that are induced under heat shock. And then we shift the cell to the normal temperature. You can see that these aggregates gradually get dissolved. And the dissolution of these aggregates involves a chaperone protein that in yeast is called HSP104 that forms a hexamer complex, use energy of ATP hydrolysis and thread these aggregated proteins through and then unfold these proteins. And then these proteins are thought to either get refolded by HSP70 type of chaperones or get degraded. So for example, in HSP104 gene deletion mutant, so these mutants do not have HSP104 encoding gene, then these aggregates cannot get dissolved. So that's what was known already. What was a surprise to us was that it turns out mitochondria also has a role in dissolving these protein aggregates. So again, this is just a little review of mitochondrial biology. So mitochondria, of course, are built up of proteins and lipids, and most of the mitochondrial building blocks, the proteins inside mitochondria that build mitochondria, are encoded by the nuclear genome, and they're made in the cytoplasm, and then they're imported into mitochondria through these pores. There are two pores. One is called the tongue complex. The other one is called the Tim complex. One is on the outer membrane. The other one is on the inner membrane. So these unfolded proteins have to be like threaded through these pores in order to get imported into mitochondria. So we found that proteins involved in mitochondrial import somehow are required for dissolution of the protein aggregates that are on the outside of mitochondria. So that led us to make a very bold hypothesis that maybe these misfolded proteins, aggregated proteins, after the chaperone disentangled them, maybe they get threaded through mitochondrial pores into mitochondria. So now, how do we actually look at that process? We want to see it. And that's actually very challenging. You can imagine if we just tag these cytoplasmic aggregated proteins with GFP, it's going to make the entire cell glowing green. We can't really see what's in mitochondria. So to get around that, we use a modified GFP system. This is called the split GFP system. So the GFP is made up of 11 beta strands forming this barrel-like structure. So split GFP cuts this into two parts. The first part has the first 10 beta strand. The second part has the 11th beta strand. And only when these two parts are in the same compartment, a fluorescent protein can be reconstituted. So what we did then is we put this large part of GFP 1 to 10, into mitochondria using a sequence called mitochondrial targeting sequence. We also link on this construct a red fluorescent protein called M-cherry. So this is a genetically coded construct we can put into the cell will express this and send this protein into mitochondria. So this is sitting there waiting, but it's not fluorescent because it's missing that last beta strand. And then we put that last beta strand on a protein that we know form aggregates. And again, this protein, when it's in the cytoplasm, is also not fluorescent, but only if it's imported into mitochondria, then the mitochondria becomes fluorescent because now these two parts can come together. So it's a really nice assay to look at, specifically looking at proteins entering mitochondria. So now here, just some examples. Here are two proteins that we know form aggregates. And you don't have to remember the name of this protein. We just basically put GFP11, this small part of the GFP on the C-terminus of these proteins. And here's on the normal condition, normal temperature, these proteins do not aggregate. There's no signal or split GFP signal in the mitochondria, which you can also visualize with this red fluorescent protein. Now when you give cells a heat shock, now the proteins start to aggregate. Now you can start to see these proteins now accumulating in the mitochondria. And this is looking at, this is a 3D reconstruction using super resolution imaging where the red is mitochondrial membrane and the green is a split GFP system, so we can nicely see these proteins are imported inside mitochondria. So now what is the fate of these proteins once they get imported? And it turns out they get degraded. So here we give cells a heat shock, you see these proteins in mitochondria, and then we shift it to the normal temperature, these proteins gradually, the split GFP signal gradually decays. And if we mutate a protease inside mitochondria called the long protease, now the decay of the signal is much slower. So this is one of several pieces of evidence that these proteins are, once they get imported into mitochondria, they're actually degraded by mitochondrial proteases. And it turns out that different proteins actually are degraded by different mitochondrial proteases. For yeast, there are at least 15 different proteases inside mitochondria. So mitochondria is actually a really protease-rich compartment for degrading proteins. So here's just a model summarizing what I've been telling you. So we made this very unexpected observation that protein aggregates are bound to the mitochondrial membrane outside of mitochondria. And after this disaggregates proteins, disentangle these aggregated proteins, we found that surprisingly they got imported into mitochondria and get degraded. And my students who discovered this pathway gave this a very cute name, MAGIC, standing for mitochondria as guardian of proteostasis in cytosol. So if you're familiar with protein degradation systems, you probably know that the cells already have two very powerful degradation systems. One is called the proteasomes, which degrades ubiquitinated protein substrates. And another one is what Mike just mentioned, autophagy, which in the lysosomal system that degrade membrane proteins and even known to degrade certain protein aggregates in mammalian cells. So how does this MAGIC fit into this picture? Why do we need this third degradation place? And the way I think about this is when I first went to America, I grew up in China. I went to US when I was 17. First time I went into an American household, I saw a really strange picture that people were stuffing trash into the sinkhole. And then, as I was horrified looking at it, they turn on the switch, and there's a huge noise, and then the trash gets ground up by a secret disposal inside the sink. So that's how I think about this mitochondria-associated degradation system. It's very counterintuitive. So I don't know if Australians have this in your kitchen, even though it's really strange, but now I have a house in the US and I love it because it's so convenient. You know, when I cook, I can throw the trash, these soft, spoilable food directly into the sinkhole. But there's a problem. It's also very finicky. So if you happen to drop a chopstick into it or forks and knives, it really messes it up very easily, and then the whole sink stops working. Okay, so that's a fear of thinking about mitochondria as a trash disposal. So then we ask the question is, what if there's too much protein trash, or misfolded proteins getting into mitochondria? So we can simulate that situation by mutating another type of cytosolic chaperone called HSP70. So when these chaperones are interfered with, there's some massive misfolded proteins in the cytoplasm. now they continuously get imported into mitochondria, even when cells are recovering at normal temperature after heat shock. Under this condition, mitochondria become highly fragmented and also accumulate reactive oxygen species. And both of these are hallmarks of mitochondrial damage. So that led us think about this. That is, we know that in age-related degenerative diseases, there are two pathologies that are almost conjoined twins. One is mitochondrial dysfunction, and one is protein aggregation. So now the question is, are they related in any way? And one of our hypotheses that what if the proteins that actually cause neurodegenerative diseases that form aggregates are also substrates of magic, and they're bad substrates like chopsticks falling into your sinkhole, and they're messing up mitochondria. So, so here we tested this hypothesis. An easy way is actually we express some of these disease proteins such as amyloid beta or tau or alpha-synuclein in yeast, and very easily we can see them in mitochondria using this split GFP system. And recently we've been following up on alpha-synuclein going into mitochondria. Indeed, we can show that this protein mess up many aspects of mitochondrial function. And recently, postdoc in the lab, Xizhang, made a mouse where alpha-synuclein can be visualized in the mitochondria of mouse, in the mouse brain. And very interestingly, we see alpha-synuclein accumulate in the mitochondria of dopaminergic neurons, which are neurons that undergo degeneration in Parkinson's disease. And interestingly, this accumulation increases with age. So old mouse have a lot more these alpha-synuclein aggregates in the mitochondria, in these neurons, and in some of the other cell types as well. So, and here's I just want to quickly mention that we can use yeast as a really powerful model system to quickly understand what are all the genetic regulators of this magic pathway. So for example, in a piece of data that we haven't published, another graduate student in the lab, Arthur Wang, working with Lin Hao, who actually discovered the magic pathway, they did a whole genome screen. So where they introduced this split GFP system into a set of yeast mutants, 4,000 yeast mutants. Each one deleted one of the yeast genes called non-essential genes, meaning that these genes, when they are deleted, cells actually do not die. So then they can ask which one of these 4,000 genes actually regulate this magic pathway. So that led them to identify over 100 genes that are really important regulator of this pathway. And interestingly, a lot of them fall into this category of regulating or involved in metabolism. And I just want to mention one of the really exciting genes involved here is a gene encoding a protein called AMPK. And this is full name is AMP activated kinase, which is known to be a master regulator of cellular metabolism from yeast to human. So when cells have only limited glucose or calorie in their food, then AMPK become activated. And a consequence of that is to induce mitochondrial genes to enhance oxidative phosphorylation, mitochondria-dependent ATP production. Because another way of producing ATP is in the cytoplasm through glycolysis by burning glucose. Now, when there's a lot of glucose, cells switch mostly to glycolysis. This is when AMPK is inactive. So we showed that when AMPK is inactive, such as when this SNF1, which is the yeast name for AMPK, is deleted, then this magic pathway is overactivated. And now if we, for example, delete this inactivating phosphatase that inactivates AMPK, or put cells under low glucose condition, then AMPK becomes highly active, now the magic is shut down. And we can show that under this condition, the cell is actually protected when there's a lot of misfolded proteins. So just to summarize, we think that this AMPK, which is a regulatory kinase that's activated under low glucose condition and also under exercise, interestingly, when this is activated, mitochondria is now focused on the oxidative phosphorylation and shut down this magic pathway for this moonlighting function in degrading misfolded proteins. Whereas under high glucose, AMPK level is low, the magic is high, and the mitochondria deals with misfolded proteins, and that's actually when mitochondria are very vulnerable to be damaged by misfolded proteins. So finally, here's a hypothesis we're trying to investigate in our ongoing work. That is, perhaps there's a mitochondrial root in neurodegenerative diseases or other degenerative diseases. And we know that there's mitochondrial dysfunction and protein aggregation that both contribute to this disease. But we now know that mitochondrial dysfunction can lead to the accumulation of protein aggregates. At the same time, if there's cellular stress that generate a lot of misfolded proteins, that can in turn lead to overwhelm mitochondria's proteostasis capacity and cause mitochondrial damage and dysfunction. Now you can think of these two arrows as a positive feedback loop or a vicious cycle that when both of them are occurring, perhaps I can precipitate disease. So we're hoping to understanding these arrows at a mechanistic level so that we can actually figure out how to stop it. And perhaps one way to do that, based on the new data I just showed you, is healthy diet, low glucose, exercise by activating AMPK can perhaps preventing damage to the mitochondria when there's proteotoxic stress and protein aggregation. Thank you. Here's my lab at Johns Hopkins, and you can probably imagine this picture was taken right before the pandemic in a tiny elevator. And this is my growing lab at NBI. This is our founding agencies. Thank you.
Thank you, Professor Rong Li. So, any questions from the audience for Professor Rong Li?
Maybe I missed it. So if in the monocell, if you degrade the protein aggregate, can they also generate another 20 or 30 passages?
Well, that's a good question. So we haven't done that experiment, but it's known if you prevent the clearance of protein aggregates, the mother will have very short lifespan. The issue is that as the mother gets older, the mitochondria become damaged. That further reduces ability to clear protein aggregates. So that's where a lot of things start to go downhill. So we have to figure out how to maintain mitochondrial health or use other artificial ways to maintain, to get rid of protein aggregates and then test what you are asking.
Thank you. Nice talk. In the 4,000 gene knockouts, that's like a very clever idea. I was wondering how you determined it from the microscopy. Did you just have to look at all 4,000? Was there any automation to help you isolate genes?
Yes. Yeah. So we took advantage of the fact is that only when these misfolded proteins are imported into mitochondria, the cell gets bright, and the signal, of course, is in mitochondria. So we can first actually use what's called high throughput fluorescence cell sorting analysis to very quickly do a quick screen of the 4,000 mutants. And then we get a bunch of hits. And then we look at under microscope one by one. Again, we can do this in semi-high throughput fashion. But there are ways to also just simply do imaging in high throughput to go through the entire genome.
So have you tried to to inject healthy mitochondria into the mother cells, or maybe take out the mitochondria from bud cells and re-inject into the mother cells. Would that reverse the aging process?
Yeah, no, so that's a great question. In yeast, that's very hard to do because yeast cells are tiny. People have done that in eggs, in mouse eggs, and even trying to do that in human eggs. Is because when eggs undergo the aging process, mitochondria also is known to become dysfunctional. So one of the therapy that has been explored is to take out the old mitochondria and inject mitochondria from young cells, and there's definitely evidence for rejuvenation. The only problem there is that mitochondria also carries DNA. So the host and the donor mitochondria DNA need to really match well, and that can be a problem to cause toxicity.
Hi, it's really nice to see this mechanism of magic. I missed from your talk, but it's also maybe a stupid question already shown in textbook. So after degrading those proteins in mitochondria, do they recycle the ingredients? And then would there be any candidate gene in your 4000 gene that's regulating this state? And when it go wrong, you will have phenotype.
Yeah, no, that's a great question. So first, whether after mitochondria degrade these proteins, what happens to the product, which is amino acids and maybe small peptides, we don't know. But it links to another question that people often ask me is that why do cells use their mitochondria to degrade proteins? Does it do any benefit to mitochondria? We don't know that. In fact, we only know that it does damage at the moment. But if you think about, again, this is speculation now, the origin of mitochondria, which are endosymbiont. They're endosymbiotic bacteria in this primordial eukaryotic cell. So importing misfolded proteins or unfolded proteins is probably how that endosymbiont eat and get nitrogen, get amino acids. At the same time, helping that primordial eukaryotic cell, which at the time probably were not very good at folding proteins, to get rid of those misfolded proteins. So it's a really nice cooperative relationship. And whether that's still beneficial to this day, we don't know. In the mutants that we've got, there are certainly mutants that are affecting the clearance step. So we can see proteins get imported and then their degradation gets slowed down. And we know some of those genes are these mitochondrial proteases that helps clear these proteins.
Second, a very short question. AMPK, do they have different activity or expression in stem cell versus the progeny of stem cells right after one round of division in mammalian system?
Yeah, I don't know the answer about AMPK level in different stem cells, but it's definitely it's a kinase whose activity responds directly to nutrient environment, nutrient availability, the energetic state of the cell.
Hello, hi. I have a question. So fantastic talk first, and then I have a naive question. So when you apply the heat shock treatment for the cells, the heat shock response gonna immediately upregulate at both gene expression level and protein expression level of heat shock proteins like HH90, HH70. I'm just curious, when those protein aggregates get into mitochondria, how do you know those proteins got degraded but not refolded or rescued by the upregulated mitochondria SP proteins?
Yeah, so those proteins we've looked at by both, you can see the fluorescence signal goes away. So if the protein is just refolded, you wouldn't expect the GFP signal to go away.
I see.
And also I didn't show all the data, of course. We've also used biochemistry to show the degradation of those proteins as well.
Nice.
Yeah.
Thank you.
Thank you.
think I would just thanks Professor Rong for beautiful presentations. So I think we'll finish all the talks today. Thank you, everybody. So we just have a little bit of time. So I think we got a beautiful panels of the plenary speakers here, as well as our lovely president, Professor Boris. So I would just have a little discussions. Maybe, Boris, you want to say something or summarize this for the entire meetings and also public lectures series?
Yes, I'd just like to emphasize the importance of mechanobiology. Given, if you think all the way back, we permanently live in a gravitational field, which is constant mechanical force. Also, life is made of living organisms are made of 70% of water, so we are permanently exposed also to osmotic pressure. So these are constant mechanical forces which we just take for granted and so on. And then on top of that, there are these intermittent mechanical stimuli from environmental. If you think in terms of plants, wind is working on them, and if you think also from, let's say, mammalian organism also touching each other, and touch is very important, as you know. Babies, I read somewhere, I don't know how true this is, but I read that babies who are being hugged a lot by their parents, particularly mother and so on, they turn out into very healthy adult people and so on, whereas it might be that the other babies who may not be so loud and so on may develop neurosis and so on. This is not really any scientific evidence, but I would not be surprised that this is the case, because touch is one very important way of communicating particularly between the social beings like humans. And and the other thing I like to add is why, we today have so many various instruments and methods have been developed so much that we can finally study mechanical force, effects of mechanical force on living organisms, which was really, really hard in the past. Because mechanical force, as I said, it's like temperature, similar, it's permanently there, present, and to study and being able to discern from that you don't record some artifact, you really need multidisciplinary approach. And this is only possible because the engineering goes head in head with the scientists, and particular basic sciences, as Marty emphasized. So that's, and it's a very growing field. I like to mention also that importance of mechanobiology may be emphasized by the fact that last year Nobel Prize for Physiology of Medicine was, one part of it was given for discovery of mechanosensitive channels which underlie touch and a lot of mechanical senses which we humans have, and living organisms in general. So that's I like just to add as a general comment.
I would just want to add on that. So Professor Boris Martinek is the founding president for the newly formed Society of Australian Mechanobiology. And the time we talk about, it is important to have this group in Australia or internationally. And there was a very, very, very interesting statement made by Boris that, it is very important when someone punches your face, you feel pain. That's the mechanobiology. And that this is really important,
that we need. I want to talk about the pain because there is a lot of pain doing science. But
yeah. And I have a question from my colleagues. Actually, they left, but then I particularly want to bring to the panel, and also I think more relevant to Professor Michael Sheetz. I just want to pass that question. So it is, in the Asian, the Chinese medicines, they talk about a lot of this magic, is so-called magic therapeutics that using acupuncture. We heard about this cupping therapies, that people actually took this this massaging or this pressure-based or the temperature-based therapeutics, and actually can boost the health and rejuvenate the body. And I personally do a lot of this cupping therapies, because it's just my family told me, but I never think about scientifically, how that thing could work. So, but there are lots of these mysterious, magic things happening around us. So, Michael, do you have some comments on, on that,
On magic?
the acupuncture, all this massaging,
No,
do you think there's scientific rules for these things to rejuvenate, reverse the aging?
I think the issue that we're becoming, aware of in many ways is that the activity and the distortion of the cells mechanically has many important implications. The acupuncture I have not studied, so I can't really comment on the details of it. However, again, one would think that those activities are selected over the years for beneficial, interventions. So, yeah.
So there's a prize given in the United States called the Golden Goose Award. The Golden Goose Award goes to science that sounds really strange, but then turns out to be very useful. And I think Boris may have actually been referring to this, not exactly the way he described it, but about 3 or 4 years ago, they gave the Golden Goose Award. By the way, GFP won a Golden Goose Award too, so I should say, state that right from the beginning. Anyway, they gave it to a group of researchers that were studying some condition in rats. And they had a particular mutant in the rat that they were interested in. And it was so hard to make it that they wanted to be incredibly careful about the babies that were being produced, the rat babies. And so they took exceptional care of these rat babies, took them away from the mothers, and gave them their own special penthouses to live in. It was all very nice. And they did not thrive. They didn't work at all. And then they looked at the babies that stayed with the mothers, and what they found is that those babies were doing all right. And they were doing all right. One of the things they noticed about the mothers is that the mothers were constantly licking the babies. And this intrigued them. And so they decided to test this. They did not lick the babies, but they did take the babies and stroke them with like a Q-tip or something like that, a swab. And they found that it was the physical touch that seemed to be very helpful in these early babies. This is an old experiment by Harry Harlow of using a terry cloth on a heat platform for newborn rhesus monkeys and stuff. So there was something about this, but what was the application, or what happened to this? Well, what they realized is that in hospitals all over the world, premature babies are taken away from their mothers and put in incubators, and they found that what really helped these babies survive was sort of a sort of massaging and careful touch to them. And so this led to a way of thinking about what happened with premature babies. So there is something with touch. There's another thing is there are some individuals. I have a sister-in-law who's an occupational therapist. And we were talking, as we often do, about various things. And it turns out that there are individuals that seem to have not as much sensitivity to touch, not lost, but not as much sensitivity. And the occupational therapist know exactly what to do to comfort them. And it's something called deep muscle massage. So again, probably stimulating mechanical receptors, although that's a guess on my part. But it is something where there is this feedback. So could there be something that in other cultures that people have come across the same thing? I mean, we all like to be hugged. Frankly, there's something that's quite nice about that. But I think that there is this aspect of mechanical stimulation doing more than just telling us there's something touching us, that there is a psychological component to this as well that's probably very important.
It kind of reminded me when I was young. When I was young, the TV doesn't work, and Dad said you just give some heat and it works. So, but it is, it works.
But not your kid.
Yeah. So I think that's a very good one. So the last one I want to particularly talk about this Professor Rong Li's topic. About the aging. I think so. Any thoughts or any ideas, suggestions that because we are facing this this is society challenge in this new century with this aging populations, is getting into a social problem. So that's why I was thinking scientifically, or from this, from your research, but also as a leadership, from your institute director, how do you think these things going to impact our future?
Yeah, so it's a great question, and that's why at NBI, aging research really becoming a theme. In part because if you think about aging, mechanical problem is really at the heart of aging. From skin getting wrinkly, from heart beating too slow, from muscle gets weak, and then to our cells, immune cells maybe are not, running fast enough to chase bacteria. So there are lots of mechanical problems on all sorts of scales. So we really can be at the frontier of anti-aging research to understand why molecular, cellular, and tissue mechanics start to fail, and how to repair them. Just like, when our cars start to malfunction during the aging process, if we understand what might go wrong, first or more easily, then we know exactly what to do to maintain and repair. So.
And then do you think, like Professor Michael Sheetz showed, there's a startup company, and you reckon there's lots of the commercial opportunities and also translational, industry moving in that direction?
Yeah, absolutely. I mean, our interest is still fundamental research, but I think that when there's an opportunity, I don't want to say that there's a contradictory between basic research and research that can lead to something technology that really benefit the society. I think our basic scientists want to do that too when there's an opportunity, when there's funding. So we just need to provide the environment and resources to allow our scientists to capture those opportunities. And that's what we're actually trying to do there.
Can I just add one thing to that? There often is, and I have to say it's often said by politicians, that there's the ivory tower scientists, the people that, and it's a false dichotomy. I agree with Rong entirely on this. Everyone I know that submits a grant proposal has to say what the implications of their work is. It's not that people just blindly go and do research and, I'll do some other research now, I'll do some other. One thinks of the consequences of the work. It's part of the application process that we all are involved in. So I don't know a single person doing fundamental research that doesn't think about his or her, the consequences of his or her results. So it's not really, it's a false statement that there are people in ivory towers that really don't care about people or don't care about diseases. There's just lots of different approaches to gaining the knowledge that we all need to be able to address these really big issues.
That's totally true. I think, do we have more questions for our panel speakers? If not, I would like to conclude the session and also thank all the plenary speakers
She
today.
seems to be raising her hand.
Oh, sorry, one last one. Sorry. Last one.
So recently I'm teaching thermodynamics, right? We all know the ultimate fate of universe is entropy. Then if we think of the cell, right, they should follow that. So as entropy gain, we can think of the information loss too, right? So I'm curious, like, is there Maxwell-Demons molecule or some function like in the cell, right? So when we are exercising, or maybe the sonography is like doing work, right? It's like the engine. But how is that transduced, like to restore the information or order? Then maybe the cell become young again. Like any possible mechanism for that, or?
I think the issue from a physicist's point of view is that they're very happy to hear that death is programmed. Because if death is programmed, it can be deprogrammed. So I think that's a challenge, but certainly it's theoretically feasible.
So I'm going to actually point to Rong, so maybe she should say, but she pointed out a good example of what happens a lot in cells. We have lots of mechanisms in cells that correct for DNA damage. We have lots of things that correct for abnormal proteins that are, so there's a lot of fail-safe methods in cells. Stuff accumulates, and that's a problem. But all the things that she mentioned in her talk are exactly the things you're asking about. Are there things, I mean, are we just all going just downhill all the time? Or has the cell evolved ways to do it? And our bodies have evolved lots of ways. One of them is cell death. One of the ways of getting rid of cancer cells is to have the cell that's going to give rise to a cancer die. So it's been said by some people that the question is not why do we have, why does cancer come up at a certain rate? It's why don't we all have cancer all the time? It's because we have surveillance systems that are taking care of these things. So the systems are there. We got 10 to the 13th cells. There's a lot of different things going on. And Rong, go ahead. Sorry.
Yeah, actually, just relate to what Marty was saying. So one just simple idea to your question is when we exercise, our cells, especially our muscle, burn up the glucose, because it, use up a lot of ATP and then drive the cells go into oxidative respiratory metabolism. And mitochondria, we've shown under that condition, mitochondria shut down, processes such as entry of misfolded proteins. So it's a protective mechanism. They get protected, and then they use all the memory potential for generating ATP. So that could be one reason that exercise can keep your mitochondria healthy, and that's really important.
And if I just can make a general comment, when you talk about entropy and thermodynamics, now that's important. Then the definition in physics is that the entropy of the closed system will lead to the death of this system, if you want. So which means basically the loss of information, increase of disorder, that's what entropy does in system. So at the first glance, living organisms somehow look like being completely contradictory to that. But we should not forget that, let's say, if you take us and the whole, just our solar system, from the plants, the sun energy goes, they generate this energy which we consume, and then in form of ATP or whatever, they keep us going. So this is, but together we are closed system. Universe is closed system, and universe will eventually die. I don't know, there are different numbers they give, some say 14 billion years, some say 40 billion years, doesn't matter. Every day, and one day it will be end of the universe. That means it doesn't matter how old we want to become and so on. They will be all dead in this universe. That's all. But this is huge time, and it doesn't matter for our even species existence. So in those terms, we can rejuvenate, and ultrasound can keep us young, and exercise can keep us young, and so on. This is all fine. So I will not worry yet about the death of humans.
Thanks. Okay, so I think let's stop by here. Thanks everybody for coming, and also thanks all the plenary speaker. Thank you.
Yes, thank you. That's, yeah.